So inhibitor is a small molecule that can prevent the action of enzymes. So this is what I've taught you the first time I introduced to you inhibitor, right? It can prevent the action of enzyme. And then over here, I actually elaborated what do you mean by prevent the action of an enzyme, right? So what does it do by preventing? It reduces the enzyme activity. Now I said enzyme activity is the rate of reaction, okay, the rate of enzyme-catalyzed reaction. So how does it reduce it? By binding to the enzyme and therefore preventing it from catalyzing its reaction. So this elaboration of point number one. So let's say, for example, the question is a 3 marks question. If you write the first point, you get 1 mark, right? So how are you going to get the second mark and a third mark is in the second point here, which you need to elaborate. So it prevents the action of enzyme. The action enzyme is it prevents the action of enzyme. Action of enzyme is to catalyze, isn't it? To catalyze its reaction, the reaction that it will catalyze. So it prevents the enzyme, right? It prevents the enzyme from catalyzing the reaction. So when it prevents the enzyme from catalyzing reaction, what will happen? The enzyme activity will decrease. Can you see that? So that you get your 3 marks if it's a 3-mark question. You see that? All right. Okay, so that's why I added into this for elaboration purpose. Okay, even if I don't add, you can get the full marks. You have to think. You have to think what I have learned about theory about the action of enzyme. What do you mean by prevent the action? Means prevent the catalyzing of reaction. And what happened after preventing? The rate will drop. Okay, the enzyme activity will drop. So you can still figure out the 2 additional points there to get the marks. Okay, but I put it in so it's easier. You learn it together. I have also, you see, I've also added some points here, right, to make the definition more accurate. So competitive inhibitor, again, I don't use the word inhibitor. So it is a small molecule that can reduce the enzyme activity. That means it can reduce the rate of reaction. Okay, all inhibitors can reduce the rate of reaction. So first statement. Next, what happened? By competing with substrate molecule for enzyme active site. This one I'm very surprised. I saw this in the new textbook. Even though in the past year mark scheme, they reject this point as an answer. That means students write this, they don't give mark. And then it happens that 2 questions in 2 different past year, 1 question they accept and they give mark. 1 question they don't accept and they don't give mark. Okay. And now I saw it in the textbook that they actually wrote it in the definition of the competitive inhibitor, which means to say from now onwards, they will accept this as a marking point. Okay, which is actually correct because competitive inhibitor actually will be competing with the substrate, right, for the enzyme active site. So it will be competing. So both of them will be competing for the active site. So this statement is actually not wrong. This statement is actually quite accurate. Okay, so I guess why in one of the past year, this is not acceptable because the examiner decided that if I use the word competitive here, if you use back the competing, you're not elaborating the meaning of this term. But this is the best word to show that both substrate and inhibitor are competing with each other for the active site. This is the best word. Okay, so therefore, when you write this point now, you should be able to get marks. Okay. Why? Because it's in the textbook. So that's why I said textbook is your mark scheme. You want to know whether you write this point got marks or not, just go to a textbook and check. If the textbook got mentioned this sentence, this point, guarantee you get the mark. Okay, so that's why I asked you to get the textbook. So what happened when inhibitor compete with substrate for enzyme active site? This will reduce the probability of substrate sitting into enzyme active site already, right? Because the chances are from originally 100% can sit in, when there's no inhibitor, becomes 50% of the chance can sit in. So reduce already. So after reduce, what happened? So it will reduce the probability of the substrate sitting to enzyme. So reduce property of ES complex form, reduce property of product forming. So therefore the rate will decrease. Okay, so can you see how you can explain in biology so that your explanation is crystal clear? Okay, because of one thing happened, the next thing happened. Because the next thing happened, the next thing happened. So it's crystal clear understanding. And there's one more point. Its ability to inhibit the enzyme depends on substrate concentration as well as inhibitor concentration, which I'll show you today. Okay, I have not mentioned this yet. So I'll show you today. Inhibitor concentration also affect ability of the inhibitor to inhibit enzyme activity. So easiest way for me to show you is this. So now in this unit volume, you have got one substrate, one competitor, one enzyme. If I want to illustrate how will the rate be affected when I increase inhibitor concentration, I can straight away increase my inhibitor inside this unit volume here. Okay, so I start to increase my inhibitor now. 1, 2, 3, 4. Okay, so now I've got 4 inhibitor here versus 1 substrate. What is the property of the substrate sitting in? Okay, which class am I having? I've got 2, 1, 2, 2. Okay, all right. So Sufyan, so what is the property of my substrate sitting into the enzyme?
1/5.
Yes, very good. Enzyme active site is actually equals to 1/5, right? Okay, because altogether there are 5 molecules trying to sit in, and your substrate is one of them. Okay, the chances of sitting in is 1/5. Now, compared to when you got originally only 1 substrate, okay, sorry, originally only 1 competitor. So when you only have 1 competitor, what is the property of substrate sitting into enzyme active site? John, 1 out of? John is not here, I think. Okay, John is not here. So what about Andrea?
1/2.
What is the probability? 1 out of?
2.
Yes, 1 out of 2. Okay, so can you see that when I increase my inhibitor, can you see what happened to the probability of substrate sitting in? From half, it has becomes 1/5. So if I continue to add more and more inhibitor, can you see that the probability of substrate sitting into the enzyme active site is definitely going to decrease already? Can you see that? Okay, so therefore, when I increase my inhibitor concentration, you will see that the rate of reaction is going to drop. Okay, it's going to drop because the probability of my enzyme sitting into enzyme, the property of my substrate sitting into my enzyme active site actually goes down, right? Okay, so with that, are you able to show me on the graph? The graph that I showed you yesterday, yeah? Give me the graph. I hope you have drawn the graph into your notes here, this graph. So if I use this graph to illustrate, say for example, this one here, I have got 1 inhibitor per unit volume, okay? Now, what about the condition when I've got, let's say, 5 inhibitors? Sorry, just now I give you 4, right? Okay, what about I've got 4 inhibitors per unit volume? So I've increased the concentration inhibitor. Where should this blue color line? How will I draw the blue color line now when you've got 4 inhibitor? Okay, Gan Yuhan.
It will not reach the Vmax, is it?
Okay, say, so Vmax. So Vmax is when it's at the region over here, right? Vmax will be achieved at this region. This region is when the substrate concentration is at the higher range, isn't it? Very high. So how high is high? Usually we use 1000 to represent substrate concentration, or maybe 10,000, okay, versus 4 inhibitors. Are these 4 inhibitors negligible? Yes, it's negligible at high. It's negligible at high substrate concentration, right? So therefore, what is the Vmax achievable?
Oh, same is it?
Same, isn't it? Same as that when there's no inhibitor, because the presence of the inhibitor is actually negligible already, because my substrate concentration is so high that chances for these 4 poor inhibitor to sit into enzyme is totally negligible, as if they're not there. Okay, so my Vmax is still the same. What about at low substrate concentration? At lower substrate concentration, where should the line be? You already know that when you increase substrate concentration, your rate is going to drop, right? So from here, if I change to here, I know that the rate is going to drop. So if my rate is going to drop, so let's say, for example, over here, okay, over here, you can always use this way to actually find out one. So over here, my blue color line is actually is here, okay, this point. So at this concentration of substrate, my rate of reaction is over here, correct? But now, when I use 4 inhibitors, I expect my rate to be lower for the same substrate concentration, right? So if the rate is lower, it should it be located up here or located down here? Up or down? Chu Jiaying?
Down.
Down, right? So I should draw the point down here, right? Anywhere down here doesn't matter, okay? So that means to say I expect my curve to always be lower than the blue color curve, but at the end of the day, it will still be touching the Vmax, right? So therefore, I'll be drawing my line always lower than the blue color line. Okay, always lower than the blue color line. And then eventually it will level off at the same Vmax, but it level off later, okay, later at a higher substrate concentration. Okay, so can you see that if I continue to increase to 10 inhibitor, where should be my line now? My 10 inhibitors versus 4 inhibitor. Is it higher or lower than the red color line? Alexandria? Higher or lower if I use 10 inhibitors now versus 4? Is my rate higher or lower, Alexandria? Is my Alexandria there? Hello? I'm expecting 17 students. Alexandria is here. Yeah, you're here. Can you answer the question? Her mic is unmuted, but she's not talking. Your mic got problem,
Can you hear
is it?
me?
Ah, yes, can hear already. Yeah. Higher or lower?
It will be lower.
Yes, very good. Lower, right? Because when you increase more inhibitor to compete with the substrate for the same active site, probability of substrate to see it becomes even more reduced, right? So the rate is even lower. Okay, it's even lower. So I use this one to represent even lower. So I'm expecting my pink color curve to be lower than the red one, right? Okay, so it's lower, 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 but eventually it will still level off at the same Vmax because 10 versus 1000 is negligible. Again, can you see that? So how do I simplify the whole curve? Very easy. I don't want to draw so many curves. I just want those standard one. I just have to draw one line like this. When I increase my inhibitor concentration, I will expect the curve for the inhibitor, the curve when the inhibitor is present to actually decrease. So please add this into your notes when you draw this graph. So it illustrated as my inhibitor concentration that I use is increasing, then I'll expect the rate to actually drop. So the curve will actually go lower and lower. But don't forget, by Vmax for competitive inhibitor is still the same, but the Vmax for the non-competitive inhibitor, is it the same or higher or lower? 极限
Lower.
Lower. Why lower? It's correct, it's lower. Why lower? When you got more inhibitor, you got more inhibitor to bind to more enzyme, right? Correct? It binds to more enzyme. So more enzymes are no longer able to catalyze the reaction. So it actually decreases the number of enzymes that are active. So when you decrease the enzyme concentration, you will decrease the number of enzymes available to collide with the substrate. You decrease the available enzyme for substrate to sit in. Okay, so therefore you're decreasing the chances of forming ES complex. So the rate will drop. Can you see that? Okay, so again, it's the same thing, but the Vmax will actually be lower. Okay, it'll be lower depending on concentration of inhibitor that you use. Okay, so this one is what you need to draw into your notes. Now, for the non-competitive inhibitor, hello, here. Now, non-competitive inhibitor, right? Okay, I've added this point also for competitive inhibitor. Okay. So I elaborated about this. The ability of competitive inhibitor to inhibit the enzyme depends on both the substrate concentration and inhibitor concentration. So therefore, elaborate further. How does it affect? Because it said it depends on, right? So how does it affect? So when I increase the substrate concentration, I will actually be able to reduce the inhibition. Therefore, the rate will increase, right? Because in the curve, when you increase the substrate concentration, the curve actually increases. And the curve is actually, the y-axis is the rate. So when I increase substrate concentration, I reduce the inhibition, I increase the rate. Can you see that? But when I increase inhibitor concentration, I increases inhibition of the enzyme, I reduce the rate. So therefore, the rate will actually drop. Okay? For the same substrate concentration, you draw a vertical line, you compare, then you know that it's actually dropped. The rate has dropped. Can you see that? The rate has dropped. Okay? Draw a vertical line to compare among the curve. So this is elaboration. Okay? So where is my non-competitive inhibitor? I also have added something inside there. So please check. So I've decided to also change the definition to make it more, increase the chance of scoring more marks. Okay. Non-competitive inhibitor. So what is inhibitor? It is a small molecule. So I don't use back the word inhibitor here. So it's a small molecule. What does it do? It can inhibit the enzyme. Okay, but inhibit an enzyme is for you to actually, for you to learn, inhibit the enzyme. But sometimes examiner will not accept this because they will say that you didn't explain the word inhibit. So then what do you mean by that? It reduces the enzyme activity. So inhibit enzyme causing the enzyme activity to reduce. So guarantee you can get 1 mark now, just in case they decided not to give mark for this because you use back the word inhibit. Okay, but you learn, yes, it inhibits. Okay, and regardless of substrate concentration. So that is the definition for non-competitive inhibitor. So small molecule which can inhibit the enzyme regardless of the substrate concentration. Okay, because it's VIP, right? So I don't care how many substrate I have, definitely I got a seat for the enzyme, from the enzyme. Okay, so that's what I have modified in my PowerPoint. There's one more thing I also write there to stress. many, but not all competitive inhibitors bind to enzyme allosteric site. So that's why in the definition, it didn't mention about that it binds to allosteric site. Okay, it's because here, majority binds to allosteric site, but some non-competitive inhibitor can also bind to active site, but it binds irreversibly. Okay. Okay, so now look at the summary of the inhibitors that you have learned. So there are two types of inhibitors, competitive and non-competitive. Now, for competitive inhibitor, where does it bind to? Yung Yun?
Active site.
Competitive inhibitor binds to where?
Active site of enzyme.
Yes.
Active site of enzyme.
Yes, active site. Does it bind to allosteric site?
No.
No. So can you see that? For competitive inhibitor, they will definitely bind to active site because their shape is similar to the shape of substrate, and therefore they can compete with the substrate for active site. So its shape is definitely complementary to the shape of active site. So over here, I want you to pay attention that all competitive inhibitor will bind to the active site of enzyme only, and it will bind reversibly. So that's why it can compete with the substrate for active site. Okay, it can bind and then it can come out. It can bind, it can come out. So they can compete with each other. Okay, but for non-competitive inhibitor, majority of them bind to the allosteric site. But there's one type that will bind to the active site and it binds irreversibly. So the substrate cannot compete with the inhibitor for the active site because inhibitor already bind permanently there. Okay? According. Does how much equals to how fast? Now say, for example, I ask two of you to dissolve the sugar into the water. Okay? So you say you use the same volume of water, okay? Same temperature of water and same concentration of sugar, okay? Same concentration of sugar. But one of you use the cube sugar in cube form, okay? And the other person, you use sugar, fine sugar, okay? Cube sugar is the sugar already made into cube lah. Okay, it's actually quite big, right? Whereas the second student use sugar that are actually in fine sugar form like this, but same concentration means same amount, okay? Same amount. So ask two of you to dissolve it in a glass of water, and the water is same volume one. Now, after they fully dissolve, is the sweetness between A and B the same? Chloe?
Yes, sir.
Sweetness is the same, right? Okay.
Yeah.
So how sweet you get it at the end is how much gram of sugar you've added in, right? How much gram you've added in is how sweet you get, isn't it? Because based on equation, how many mole of glucose in solid form? Let's say how many mole of glucose, okay, in solid form? Can write like this. I don't want, I type can, okay, type better. How many mole of glucose in solid form? When you dissolve in water, you also get n number of glucose, right? In aqueous form, right? Dissolved in water means surrounded by water molecule. So can you see that how much glucose dissolved in solution has to do with how much glucose you added in? Because if you add n mole of glucose to the volume of water, you also get back n mole of glucose that's totally dissolved. Okay? Now, my second question is, so I've talked about how much, right? How much product you can get actually depends on how much substrate that you use, isn't it? Okay? Now, second question, how fast I will get my product B? Does it depend on how much A I use? So you compare between A and B now. How fast I'll get all the sugar dissolves in the water for A and B. So I ask you all to throw the sugar inside there. Don't stir. Just leave it there. Okay? Which one will dissolve faster, A or B? Sarah.
B.
B dissolve faster, right? Okay, because B has got larger surface area to volume ratio, so it will dissolve faster. The surface facing the water is small, so it dissolve faster. So can you see that? How fast a reaction takes place has nothing to do with how much you have, and how much you get has nothing to do with how fast you get. Okay, so this is a very important thing that you need to know. So fill in the blank. How much product can be formed depends on what? Pei-Wen?
Amount of the reactant.
So the mole of the reactant, very good, which is actually the mole of the substrate, right? So the mole of substrate is also dependent on what? Based on the formula, mass is equals to mole per volume. So if I ask you, sorry, actually, this is not correct, not mass, concentration. Concentration. How to use concentration? M, M. Or do you use M for concentration? Capital M, okay? So it depends on number of mole of substrate. That means it depends on the concentration, right? Based on the equation. If it depends on the mole of substrate, that means it depends on concentration of substrate, right? What about, I said, I don't want you to talk about substrate. What about the volume? Does the volume affect or not? Volume affect or not? Yes, right? The volume of substrate also affect, isn't it? Because you bring the volume over to here, also give you the mole, isn't it? So how much product can be formed actually depends on mole of substrate based on the equation, which is also proportional to substrate concentration based on the equation, based on the formula, based on this formula, okay? Also depends on volume of the substrate, also based on this formula, okay? Now, how fast a product can be formed depends on what? Yun. Give me your answer. Cepat.
If you mix it and what?
Huh?
If you mix the mixture or not.
So mixing, is it?
Yeah.
So whether you do the mixing to the solution, yes, of course you mix, they dissolve faster for this particular reaction. Okay, so other than mixing, what else can also affect how fast you can get the product? Gan Yuhan. Cepat.
The temperature.
Very good. What else, Rebecca? Cepat.
Enzyme
Enzyme what?
concentration.
Yes, enzyme concentration. Very good. Some more. Tan Jia Ying. Cepat.
Concentration of substrate.
Very good. One more. Cepat. John. Master. Cepat. Hello.
Hi.
What else?
The concentration
Concentration of what?
substrate.
Concentration of substrate already given. There's one more concentration you can mention here. What is it?
Enzyme.
Enzyme given already.
I don't know. Concentration
Inhibitor.
of
I just mentioned just now. Yes. Concentration inhibitor. Now actually there's one more. Who wants to try? Sufian, cepat.
Is it size of the substrate thing?
Size of substrate means you're talking about the surface area to volume ratio, right?
Yeah.
Size or surface area to volume ratio. This is when you're talking about only if you're talking about dissolving it, dissolving it, okay? Then you can talk about size. But if you're not talking about dissolving it, then we don't talk about size, understand? So the mixing, you see not, the mixing and the size have to do with when you want to dissolve only. You talk about it. Remember the factors affect the rate of enzyme-catalyzed reaction. You learn about temperature, enzyme concentration, substrate concentration, inhibitor concentration. Got one more missing. What is that? Tan Jiaying. Cepat.
pH.
Yes, I'm waiting for pH. My neck very long already. Like how you wait for your COVID vaccine, right? I will wait until the neck very long. Okay. Have you become giraffe already? Okay. I've become half a giraffe already, waiting for my vaccine until I get it first dose. Okay. So these are all the answers given just now, so need to see it. Okay, next move on. So you see in this series of reaction, why I call it series of reaction? Series of reactions is because a reaction is made up of the substrate converted to product is called a reaction. So this one reaction catalyzed by this particular enzyme. And then I have got a second reaction whereby B becomes my substrate converted to C. So this is a second reaction here, B converted to C. And I got a third reaction here where the C becomes a substrate converted to D, the product. Can you see or not? So I got three reactions here. So therefore, I call this a series of reactions that are connected to one another. So this whole series of reaction from A to form B, B to C, C to D, the whole thing is called a metabolic pathway. So metabolic pathway is made up of a series of reactions that are related to each other. Okay, so remember what is a reaction? What do you mean by a series of reaction? What do you mean by metabolic pathway? Our aerobic respiration in a cell is a metabolic pathway. It is made up of a series of steps before the glucose is fully oxidized to CO2 and water and ATP and heat. Okay, why you need to take up so many steps? Why not one step? Because you imagine from glucose having 6 molecules of carbon to become 1 CO2 and 1 water. You cannot one step chop off so many covalent bond, right? Because every step the enzyme can only maybe chop off 1 bond. Okay, chop off 1 bond. So therefore, you need a series of steps to finally chop off all the carbon out from the glucose to become CO2. So that's why you need many steps. It's not one step, you do everything, right? But nowadays, you've got this thing called what? Thermomix. You know, Thermomix in the kitchen. Okay, one thing can do everything for you. But the enzyme is not Thermomix, okay? The enzyme is the traditional O is gold. One enzyme do one work only. Okay, you open up your toolbox. You got many tools inside there, right? You got your hammer. Hammer is to do what? To ham. To ham the nail. Okay, it cannot screw the nail. You want to screw the nail to use screwdriver. So that's our enzyme. Okay, not one thing do all. Okay, that's enzyme. Okay, very fun. Okay, let's move on. So what I want to illustrate here is sometimes in a body, right? When there's enough product being formed, so the product here, the end product of this series of reaction is D, right? When we have enough product formed, we no longer want this process to take place anymore because I got enough product already. So I don't want A to be converted to B to be converted to C already. Okay, so therefore I have to stop all this metabolic pathway, right? I have to stop the entire pathway. So in order for me to stop the entire pathway, the best way for me to do is, of course, to inhibit the first enzyme. Because when I inhibit the first enzyme, I inhibit the whole pathway, right? Because A can no longer be converted to B. So therefore, there's no more B being formed. So when there's no more B, it will not be converted to C because no more B. So there's no more C and there's no more D already. So you stop the first reaction in the metabolic pathway, you stop the entire metabolic pathway. Why? Because you have enough D already. For example, after a meal, right? Your digestion, you digest carbohydrate to become glucose. You got a lot of glucose in your blood. So high blood glucose level. So your body quickly secrete insulin so that your liver will take up the glucose and convert it to glycogen and something else, right? So therefore, let's say D is insulin. But once your liver take away the glucose from your blood glucose level becomes normal. You cannot afford to continue to produce your D already. Then you will make your blood glucose level so low that you will pengsan. You will faint, right? So you have to stop the synthesis of insulin already. So you have to have the mechanism to stop the production of something when you have enough of that in your body, or when you no longer need it, you have to switch it off. You have to switch off this process by inhibiting the enzyme. So usually we can use this end product to inhibit the first enzyme in the series of reaction. So this is called end product inhibition. So when enough enzymes are produced, the metabolic pathway can be switched off by the end product inhibition. So this process is called, this process here, okay, is called the end product inhibition. End product inhibits the first reaction in the metabolic pathway. So usually this enzyme, I ask you to choose, is it competitive or non-competitive inhibitor? Now, usually this enzyme is a non-competitive inhibitor. Non-competitive, usually this enzyme is a non-competitive inhibitor because it will sit to the allosteric site, because its presence must inhibit the enzyme regardless of whether you got a lot of substrate or not. I must inhibit the enzyme. So I don't want to compete with you. Okay, when I've got too much of this, I have to inhibit the enzyme immediately. I don't care whether you got a lot of A or not. So it will sit into the another site other than active site to immediately inhibit the enzyme. And then when I don't have enough D, the D can actually come out. The D can come out to supply my body with D. So when it come out, then A can sit in to form B, B can form C, C can form D. So when I don't have enough D, D will come out so the enzyme becomes active again, so that this pathway is activated to produce more D for me. Okay? All right, so this is illustrating end product inhibition, where the product becomes an non-competitive inhibitor, becomes a non-competitive inhibitor. Okay? So this is an example of a metabolic pathway in a body where threonine is converted to isoleucine. So these are all amino acids, okay? So once I got my isoleucine produced already, enough already, I will switch off the series of reaction by inhibiting the first enzyme in the series of reaction. So this end product inhibition, okay? So let's try this question. It's a past year question. 2 enzyme experiments were carried out. The first experiment is X, was carried out at a constant temperature, 37 degrees Celsius. Okay. And during the second experiment, temperature was increased from 38 to 80 degrees Celsius. Okay. So, and then which graph shows the result? So first thing you must check, the graph is graph of product versus time. That means product concentration must increase with time, right? Okay. So I've got X is when temperature is 37 degrees Celsius. If you look at all of them, they look the same one. Okay, all the A, B, C, D for X is go up, the products. Okay, product concentration will increase with time as more and more substrate converted to product, and then it will level off. Okay, when all the substrate has been used up and all substrate has been converted to product. So for the second curve at 80 degrees Celsius, which one do you think is the answer? Type your answer in the chat. Wow, you are very fast, huh? B. Okay. You think again, Think again. If you say it's B, right, that means they're telling me, oh, my glucose dissolved already, and after that, it becomes solidified again. Can Hello? My glucose dissolved, after that, it's solidified. See, oh, as time passes by, I got more and more glucose dissolved, and after that, my temperature too high, so my glucose becomes solid again. So my product is gone, my substrate come back again. Cannot, right? Cannot. Once your product is formed, forever you have the product there. It will not change one. Okay, so therefore, D is also not correct. How can you start off with, oh, originally all my glucose is dissolved. You start with glucose not dissolved, right? After that, it becomes solid. Cannot, right? So D is also cannot. So you're left with A and C. Which one is correct? A or C? Did anyone write A or C just now? When I'm very relaxed, you can learn very well, but I cannot finish my whatever I plan to finish today. I'm very stressed. Hello, A or D? A or C? Okay, let me erase the whole thing so you see clearly. You look at A, is A logically correct or not? When I use a higher temperature, I expect the kinetic energy of my substrate and enzyme to be higher compared to when I use 37, right? Which means to say the rate of collision between enzyme and substrate is higher. The rate of effective collision is higher. The rate of ES complex form is higher, right? So therefore, the rate of product formation should be higher. So the rate is higher means the gradient of the curve should be higher. So can you see the gradient of curve at the beginning is higher? Can see, it's higher than X. But then because I use higher temperature, as my enzyme, okay, the atoms in my enzyme start to vibrate vigorously, then there would be cross links that possibly be broken due to high temperature, isn't it? So what are the cross links between the R group in the enzyme that can be broken because of high temperature? Alexandria? Other one, Amar, because I've not called him today. Amar, what are the bonds that can be broken by high temperature?
Hydrogen.
Very good, hydrogen bond. So what do you think is the answer now? A or C? I think A is more correct, right? So after that, it slows down because my enzyme denature already. So when all my enzyme denature, it can no longer convert whatever substrate left to product. So whatever product is produced will remain there. Whatever substrate is still there cannot convert it to product. So you cannot get this maximum amount of product that you're supposed to get from the amount of substrate that you start off with. Okay, so therefore, for enzyme chapter, first you must know the basic very well, and then please go and practice using past year question. Can you see that or not? Right? Learning theory and doing question, two different things. Can see or not? Okay. So now we go to the last one, which I'm supposed to cover today. So I cover in 5 minutes. Why? Because actually can cover in 5 minutes because this is very easy. So immobilized enzyme. So in the learning outcome, it says, investigate enzyme activity means investigate the rate of reaction of enzyme that is immobilized in alginate versus the same enzyme that's free in solution. So what do you mean by immobilized in alginate? And then state the advantages of using immobilized enzyme. Okay. Shall I record as separate?